How does p53 transactivate or turn on other genes? One of the other genes that p53 can turn on is p21. p21 is a gene that will cause the cells to stop its cell cycle. In other words, it causes the cells to stop dividing. By activating p21, p53 has helped reduce cell growth, thus making it less likely that a tumor will form. Let's look at the activation process. Any gene that is activated by p53 will have a region near it that allows for p53 to bind and start the activation process. We call this the p53 response element, or p53RE. Here is p53. As you all have learned, in order for it to bind correctly to the DNA, it first must form a tetramer. That just means that it has to bind with 3 other p53 molecules. Once this happens, it can bind to the p53RE. Now that it's bound to the DNA, it recruits or attracts RNA polymerase. The RNA polymerase then reads the gene, in this case p21, and creates an mRNA transcript. This line with the arrow indicates that the mRNA transcript has been made. What you just saw is transactivation. In the lab, measuring the amount of p21 that has been produced is difficult. There's no easy way to tell if this mRNA transcript was made. So we will use something called a reporter gene to measure how good p53 is at activating our gene of interest, in this case p21. This is how it works. We will take the p53RE associated with p21 and put a reporter gene next to it. The reporter genes we will use lead to a visible color change. In this simplified example, let's say our reporter gene produces a blue color. Now, if p53 is able to transactivate p21, we will know because the blue color will be produced. Let's say instead we have a mutant p53 that doesn't bind well to the DNA. We will be able to detect this because it won't be able to bind to the p53RE, and so we won't get the blue color. p21 is just one of many genes that p53 is known to transactivate. We can use the same method to test how well p53 is at turning on other genes as well. For example, BAX is another gene that is normally transactivated or turned on by p53. To test how well a particular mutant p53 is at activating BAX, we can use this same reporter gene to test how well p53 can activate BAX.